I'm Leslie Blankenship-Williams. I have a doctorate in biology, and I teach college-level biology courses to allied health professionals, such as nursing majors and physician's assistants. I also work as an embryologist in an IVF laboratory. As we explore our conundrums in human reproduction, we're going to turn our focus to the IVF process. IVF stands for in vitro fertilization. It has been around since 1978 when the very first IVF baby, Louise Brown, was born. IVF has gained social acceptance as an acceptable way for families to have children in the United States. And in fact, IVF was responsible for about 2% of all babies born in the USA in 2018, which is the most recent year that we have stats for. That said, there are aspects of the IVF process that certainly deserve further scrutiny and lend themselves to ethical dilemmas and/or controversies. These include, but are probably not limited to, selection of embryos for transfer. When you get ready to pick an embryo to transfer to a uterus with the hopes of implanting and becoming a baby, there are choices because we do genetic testing. So what types of choices should be allowed? Should we be allowed to choose the biological sex of the embryo that is transferred? Should we be allowed to factor in chromosomal abnormalities? What about genetic disorders? What about whether or not an embryo has a gene for an athletic build, or a high IQ, or a hair color that is desired by the intended parents? So like many things, it's not a black and white answer. My goal is not to impress my own feelings about the matter onto you, but rather to expand your knowledge base and your social awareness of these ethical considerations. Likewise, when you go through what is called an IVF cycle, very often there are additional embryos that are unused at the end. And what do we do with them? And this ties directly into this question of when does life begin, and the intersectionality between reproductive rights and looking at the future or potential future of a viable embryo. So these are all really interesting questions that don't necessarily have clear-cut answers, but as part of your goal to be a holistic, knowledgeable, compassionate, and empathetic healthcare provider, I think it's important that you have a broad and thorough understanding of this process. So we're going to just look at the IVF process here, and then in the next video, we're going to talk about the ethical dilemmas that are embedded into that IVF process. So in vitro fertilization, this is an image of me looking at a microscope, and I'm looking at an embryo on the screen. My teacher, yes, I had my own teacher, Deborah Venier, is looking at this embryo with me. Now, how big is that embryo? If I were to hold my petri dish and look down and try to find the embryo with my naked eye, I might see a speck, but chances are I wouldn't see anything. That's how small it is at this stage. This represents like a day 5 embryo, an embryo that is 5 days after fertilization. And I'm actually looking at it under a really interesting microscope called an inverted microscope, because the objective lenses are not facing down like in a typical microscope, but are rather underneath the stage looking up, which is kind of cool. You might notice there's some joysticks on the side. I'll actually describe when those joysticks are used in some of the procedures that I'm going to introduce you to. Let me give you just an overview of in vitro fertilization, and then we'll look at each one of these steps in a little bit more detail. So in vitro fertilization means that you're fertilizing outside of the human body, which means that the eggs and the sperm have to be outside of the human body. Getting the sperm outside of the human body is pretty easy. You simply ask the man to ejaculate, and as long as there's viable sperm in there, that's the end of that story. Getting the eggs outside of the body requires an invasive surgical procedure that is most commonly known as an egg retrieval or an oocyte retrieval. And so this is where a needle basically goes up through the wall of the vagina to the ovaries and aspirates out the eggs. So the eggs are recovered, the sperm is recovered, and then you put them together and fertilize outside of the human body. The fertilized eggs are then allowed to grow in culture for 5 to 7 days, and we hope that they develop into embryos very similar to the one on the screen. And then the embryo ultimately is transferred back to a receptive uterus with the hopes that the embryo implants or grabs on and creates a baby. So let's take a look at these steps in a little bit more detail. Most of the pictures I'm going to show you are actually from my training program, including this one on the screen, because if I were to go into a real IVF lab, I'm showing you real patient information and proprietary procedures that would be highly frowned upon without a lot of permissions, which I do not have. So instead, I'm showing you lots of pictures from my training program. So let's do this. So let's start with what happens typically in an ovarian cycle. During each cycle, which lasts about a month, multiple follicles are going to be recruited, and each follicle contains an immature egg. Now, these follicles that are recruited for a particular cycle, which again lasts about a month, are going to start competing with each other for limited resources. Now, what limited resources are those? Hormones, as it turns out, including follicle-stimulating hormone, or FSH. So typically, all of these follicles that are recruited in a given cycle, and the number, by the way, can range from just a few to over 80, engage in a fight to the death where only one is left standing, kind of like the Hunger Games. So I think of this as the follicular Hunger Games. The dominant follicle, or whatever the winner is, usually takes the lion's share of the limited hormones, and that effectively starves the other follicles to the point of where they die out. And so, if this process works as it's intended, only the dominant follicle actually finishes the maturation process and releases an egg somewhere around day 14 of the ovarian cycle. And the releasing of that egg is known as ovulation. In IVF, we want more than just one egg. We want as many healthy eggs as we can possibly retrieve. So we use hormonal manipulation to bypass the Hunger Games and try to ensure that there are plenty of hormones for all of the follicles recruited for that cycle, so that most develop, survive, mature, and produce good eggs. So IVF bypasses the Hunger Games, and if done well and correctly and there's a good response to the extra hormones, we can have 5, 10, 15, 20, maybe even 30, 40. We don't usually want more than 40, but you can have that many follicles that yield eggs. But we don't want the woman to ovulate all of those eggs because we want to be able to capture them and manipulate them in the laboratory. So a surgical procedure known as an egg retrieval is scheduled, and it's timed to occur a few hours before ovulation, and it really is that close to the window where you try to get it right before potential ovulation. It requires 2 physicians. You have your reproductive endocrinologist, and then you also have an anesthesiologist as the patient is intubated during this procedure. The procedure uses an ultrasound-guided aspiration, so there's a needle equipped to this transvaginal ultrasound where the needle goes up and aspirates whatever is in the follicle. It turns out that the follicle has more than just the egg. There's a lot of fluid, there's a lot of other cells called granulosa cells, and there's quite a bit of blood usually as well. So when this fluid gets aspirated, it's more than just the egg, but all of that stuff goes into a test tube, and then it is the embryologist's job to be able to find the eggs amidst all of that cellular debris and fluid. And so what the embryologist is looking for is something like this. This is called a cumulus oocyte complex. Oocyte is just a formal name for an egg. You might notice here that the egg itself is actually kind of hidden in this mass, and you get kind of a trained eye to look for it. Here we have an embryologist that is looking at a petri dish filled with fluid, and you can see what is projected up over the screen here. That's what she's looking at. And she's trying to identify these cumulus egg masses. There's actually quite a few of them in here, but you can see, just based on what I'm showing you, how difficult it can be to actually identify them. It's not easy. Once the embryologist locates and separates out all of these cumulus egg masses, they are stored in an incubator at 37 degrees or body temperature for a few hours, at which point insemination is scheduled to occur. We're going to look at a special type of insemination called ICSI. I've actually already shown you a video of that, but I'm going to show you another one. And if you are going to do ICSI, you actually want to strip away all of these cumulus cells so that only the egg is left. So this process is called stripping. Yep, you've heard that right. It's called stripping. And the eggs that are no longer protected in this cumulus mass are called denuded. We like all those terms, naked eggs and stripping. But there's the egg cell. This is what's known as a polar body. Really fascinating, but I'm not going to get into that. And this is a glycoprotein shell known as a zona pellucida that kind of protects the egg. Meanwhile, sperm is going to be purified and prepared for the ICSI procedure. So this usually involves a semen sample. And this video right here actually shows purified sperm. And this is a really good sample, by the way. Most of the time, we don't get samples that are of this high quality. But the goal is to get a good semen sample, purify the sperm, add it to a petri dish, add the eggs to the petri dish as well, and then inject the sperm in a process known as ICSI. Now, I mentioned that that was a good sperm sample, and it really is a good sperm sample. Recently, global news outlets have reported a study that showed that sperm counts are declining worldwide by as much as 50%. But the reality is that I've seen more than I'd care to admit of semen samples where the sperm counts were very, very low. They were mostly dead sperm. If they did move, they didn't move well. And that would generally be an infertile male if we didn't use the special technique called ICSI. So I think although sperm quality and quantity tends to be taken for granted when we talk about infertility and IVF, I think we're going to see more focus on sperm in the future, especially given the recent study results. An ICSI procedure stands for intracytoplasmic sperm injection. This is where an embryologist uses a micromanipulating tool, some joysticks, and basically injects a single sperm into each egg. We've seen this in a prior video, but let's watch it again here. We're injecting that one sperm in, and we're done. Once the eggs are injected, they are transferred to a special incubator that is designed to keep them at the correct pH, the correct oxygen concentration, and temperature levels that would mimic the female reproductive tract. The culture dishes for embryos is actually pretty interesting. A drop of special media formulated for the growth of the embryo, including specific sugars and amino acids, are placed on a small petri dish and then covered with oil. As oil and water don't mix, this keeps the drops of media separate, but also prevents their dehydration. So this is a culture dish here, and you can see the little drops, and they are very small. It's hard to see that this whole thing is covered with oil. If you can't quite see a drop, I'm going to highlight it for you in red. And we actually place the embryos under oil, so there's an oil overlay. We place the embryos under oil, and then we move them to an incubator with that is kept at very specific parameters. Now recently, we've started to see the use of time-lapse incubators in the IVF lab. Most IVF labs as of 2022 don't have these, but we were fortunate enough to be able to get one on loan at the training facility. So here's my teacher, Debbie Venier, with a time-lapse incubator, and she is looking at a 2-cell mouse embryo in a media droplet. Now this particular incubator takes pictures every 5 minutes, and so you get a time-lapse video of the growth and development, and we're going to watch one. Again, this is a mouse embryo, but humans develop more or less the same way. So you can see here that it starts with 2 cells. Two cells are going to divide into 4 cells. You'll notice again that outer shell, that's called the zona pellucida. Notice the embryo doesn't actually increase in size during this stage. I'm going to get 8 cells. Rather, the volume stays the same, but the number of cells increases. Now, although the number of cells are increasing here is probably about 10 or 12, they start to lose their clear delineation of boundaries, and you can't really see where a cell begins or ends, so it looks like one mass, but there's actually a bunch of cells there. It's just not clear to our eye, and at this stage it's called a morula. Now a sodium-potassium pump is going to start causing fluid to flow into the embryo, creating a fluid-filled space known as a blastocele. And that's actually going to push the embryo out and cause it to expand. And you'll notice all of the dynamic stuff that's going on. It's not a static process. There's a lot of trial and error. It tried and then it failed. And eventually the embryo is going to hatch out, as shown here. Now, in a patient's cycle, there may be anywhere from 1 to 50 plus fertilized eggs that are cultured with the hopes of growing into a full-fledged blastocyst. A blastocyst is the name that we give to the embryo when it's in this stage. I say hope because the development process often arrests or fails, and that's something I'm going to discuss in the next lecture. Only blastocysts that have developed fully with good morphology are selected to move on in the process. The rest are discarded as they are non-viable. How many zygotes actually fail to develop, as I showed you, into viable blastocysts? A lot. Sometimes all of them. But in a good cycle, maybe 50% develop properly. So now we get to our next step. We've cultured our embryos, they've grown into a blastocyst, which should look something like this, if done correctly. And now we are going to biopsy this embryo. Now, not all IVF cycles employ this next step, but most clinics will at least offer this as an option, and many clinics mandate this. So this next step is called embryo biopsy. A blastocyst, which is where an embryo will be at day 5 or 6 after fertilization, has already started differentiating its cells. We have a trophectoderm, which I have in kind of a mustard color. The zona pellucida, I mentioned that previously, that's actually a glycoprotein matrix that is going to protect the embryo as kind of a shell of sorts. And the cells in the center are called the inner cell mass, or ICM. The fluid-filled space is the blastocele. So the zona pellucida is a non-living glycoprotein-rich matrix analogous to a shell, and it contains and protects the morula and blastocyst during the early embryo development. But eventually that embryo is going to have to hatch out of that shell, and we saw a little bit of that towards the end of our video in the prior slide. The blastocele, which is a fluid-filled space inside the embryo, continues to accumulate fluid by way of the sodium-potassium pump, and this allows for the expansion of the embryo. The clump of cells here that are actually very hard to discern as individual cells when you look at a real embryo picture is known as the inner cell mass. This is potentially one of the most important components of this embryo because these cells are the cells that give rise to the fetus and the baby. The outer layer of cells here are known as the trophectoderm. Troph means nourishment, ecto means outer, and derm means skin. So this layer of cells is akin to an outer skin that provides nourishment. What could that be? Well, the placenta, of course. So if successful, the trophectoderm's job is to hatch out of the zona pellucida and attach to a competent uterine endometrium and develop into the placenta. And the placenta is responsible for facilitating the exchange between the mother and the growing embryo and fetus. Although the cell types are in fact differentiated, the ICM and the trophectoderm, they all derive from the same fertilized egg. So the idea is this: if we sample a few of the trophectoderm cells, we should be able to run a genetic profile on those trophectoderm cells, which should reflect a genetic profile of the rest of the embryo, including the all-important ICM, which gives rise to the actual fetus. So the most common way to do this as of right now is a biopsy of the trophectoderm, and the goal is to get genetic information about the embryo to determine the likelihood of whether or not the embryo can result in a healthy baby. So there's my schematic. I actually made this drawing over here. Here's the real picture. So in the real picture, the ICM is over here. The trophectoderm, again, it's not nearly as nice and pretty as what I've drawn. The zona pellucida is out here. And you can see that some of the trophectoderm cells are being aspirated, and you're eventually going to kind of separate those cells in a biopsy and analyze their genetics, and hope that whatever this tells you is reflective of this right here. So let's go ahead and watch a video. This is actually a true human embryo biopsy. This is not a very good quality embryo. This was a very difficult embryo for me to biopsy. The ICM, I don't really see it here, but I think it's going to be somewhere over here. We're going to aspirate some cells. You try to pick an area as far away from the ICM as possible because you do not want to damage the ICM in any way, where the trophectoderm is a little bit more expendable. So we're going to pick some cells over here. And I'm not a super fast biopsier, so I'm going to go ahead and speed up this video faster than it really is, just so that you're not bored, because I do play around a little bit trying to get it just right. The bullseye that you see here is actually the laser. And what I'm going to do is I'm going to start lasering some of the cells around the part that is aspirated into my tube there to separate it. You might notice there's no zona pellucida around this embryo. This embryo was already completely hatched. These are some of the more difficult embryos to biopsy. I'm starting to laser, and you can start to see the piece separate with the laser. Alright, so there's my biopsy piece. There's the rest of the embryo. Here, we can see another embryo with the zona pellucida actually still intact here. This is a mouse embryo that I'm practicing on. And part of the trophectoderm has been aspirated out there. You can see that. Lasers, and you can see the dot here is the laser, was used to help loosen and separate the cells from the rest of the embryo. The fragment, usually containing between 6 and 10 cells, is going to be removed from the solution and sent off to a genetics lab to be profiled. The remaining embryo, which is actually shown here, still in the zona pellucida and still containing its intact inner cell mass, which has sustained some damage. I mean, this is not risk-free. We do damage the embryo a little bit doing this, but the damage is usually repaired, is now going to be frozen as we await the genetic results of that biopsy. I use the word frozen liberally, because if we were to take an embryo and freeze it, the water in the embryo would actually form ice crystals. Ice expands, and that actually creates nanoscopic shards that literally shred the embryo, much like if you've ever put water in a glass container and then put it in the freezer. The water expands, the glass explodes, and you have glass shards everywhere. So just freezing the embryo like this, if you just take the embryo and put it in the freezer, like you freeze your food or an ice cube, will kill the embryo. So we use the term freeze because people like that term, they understand what that term means, but the real term is vitrification. Vitrification is also going to use supercooled liquid nitrogen. LN2 stands for liquid nitrogen, that drops the temperature below negative 200 degrees Celsius. But again, if we just take the embryo and submerge it into liquid nitrogen, ice crystals will form and destroy and kill the embryo. So how do we prevent this? The answer is twofold. We need to actually modify the water content in the embryo itself by removing as much water as possible. If we remove as much water as possible, then we prevent water from potentially making hydrogen bonds and forming ice crystals. But the other thing that we need to do is we need to add cryoprotectants. And cryoprotectants are going to be small molecules that interfere with water's ability to form hydrogen bonds in the first place. This is kind of similar to how you might add salt to water to prevent water from forming ice. So you hear about people who live in colder climates salting their pathways, and that causes the water to melt. What that's really doing, the salt is actually interfering with water's ability to form hydrogen bonds and form ice. And in the same way, we're going to introduce small molecules. They're not salts, but they're small molecules that prevent water from forming hydrogen bonds. So this is a great picture that shows if I were to just take a droplet of fluid that did not have cryoprotectants and then put them in liquid nitrogen, I would form a bunch of ice crystals and it would be opaque. If I were to add a bunch of cryoprotectants, the water molecules that are present in that drop have a lot of difficulty forming hydrogen bonds and forming ice, and therefore it stays in kind of a glass-like state that does not have a tendency to expand and shred the embryo. So I personally think vitrification is ingenious, but the science of how that works is kind of beyond the scope of this one lecture IVF overview. But suffice it to say that vitrification involves two things. We need to introduce cryoprotectants, which are small molecules that prevent water from forming ice, into the embryo cells, while also removing as much water as possible osmotically. And so this entails moving the embryo through different vitrification solutions to achieve that goal. And you can kind of see that here. This is an embryologist who's, again, doing this under a microscope, and these are different vitrification solutions, and the embryo is going to be moved from one solution to another. Once the embryo has had sufficient time to absorb the cryoprotectants and reduce the water percentage, the embryo is what is called loaded onto a very thin straw-like device in a tiny drop of this vitrification fluid. And this device is then plunged into liquid nitrogen and stored indefinitely. So this is me. I'm working again with a mouse embryo. This is a device under a microscope. It is visible to the naked eye, but I've got my embryo basically right there, and I'm going to put it in a tiny little drop on this device. I don't always have the steadiest of hands, so it can be hard to do. And you can't really even see the embryo in there. It's so small, but the embryo is in fact in that drop. There's the embryo right there, in this tiny little drop. And then that's going to get plunged into liquid nitrogen. And so here's a picture of another embryologist using a bath of liquid nitrogen and basically putting that device in liquid nitrogen to supercool it and vitrify the embryo. And just for fun, this is a nice picture of me and my friend and fellow embryologist Ryan as she is vitrifying, I think, 200 plus embryos here late at night, and I'm there doing my own work, my own research, and we're just providing each other moral support. So the next thing that happens is an embryo is selected for transfer. Usually the genetic profile comes back, you determine which embryos are good for transfer, and you select one to transfer to a uterus. For obvious reasons, I'm not going to be showing a picture of a real embryo transfer. Again, patient sensitivity there. But instead, I'm going to show you the simulations that people use to train on in what is known as a vagina in a box. So there's our vaginas in a box. There's 5 of them in this image. The embryo that is chosen for implantation is usually thawed a few hours before the transfer to let it come out of its vitrification state, come out of hibernation, and kind of regain its metabolic function and get ready for implantation. The embryologist is then going to suck up the embryo into a small flexible catheter. So there's the small flexible catheter that, or kind of sort of, not exactly that, but it looks just like that the embryologist is going to put the embryo into. Using ultrasound for guidance, the physician is then going to insert that catheter into the cervix. So this is the ultrasound guidance. You'll be able to see it on the screen. The catheter has a special tip that allows you to see it. You can see the catheter go past the cervix into the uterus, and then the embryo is deposited there. It's kind of a nice picture, nice video here, showing that cervix. So you get a picture going right in and looking at the whole of that cervix. So if all goes well, the embryo implants and yields a positive pregnancy test less than 2 weeks later. And from there, the patient is transferred to their OBGYN for continued monitoring, which of course we hope results in a healthy baby. So I hope you have found this brief introduction to current IVF procedures fascinating. Although IVF itself is now generally socially accepted and not considered controversial or taboo as it once was, components of the IVF process certainly have conundrums. In our next lecture, we're going to explore 2 of the topics that I find the most difficult. Genetic testing of embryos yields genetic information, including the chromosomal composition of the embryos, including the sex chromosomes, which remember will determine biological sex, and can also be expanded to also look for hereditary diseases, genes associated with higher risk of disease, and potentially even things that are aesthetic, like athletic prowess, skin color, eye color, etc. Moreover, there is a new technology called CRISPR. It's not yet ready for prime time, but may be soon. And this can allow us to potentially edit the genes of a zygote. So not only do we need more concrete ethical guidance about how we select embryos and how much genetic information do we reveal and what do we do with that, but also the question of editing, genetic editing of embryos, potentially to create designer babies of sorts, might also be something that we need to grapple with sooner rather than later. In addition, the unanswerable question of when does life begin, and reproductive rights closely intersects with many aspects of IVF, including, but certainly not limited to, the fact that many, and I'd argue even most, embryos fail to develop, usually because of genetic abnormalities in the embryo. But if one believes that life begins at conception, and laws are written to say that life begins at conception, and therefore a zygote has personhood rights, and these embryos fail to develop, then what? Is this manslaughter? Murder? Moreover, IVF is an incredibly complicated process designed to allow a person or a couple or intended parents to expand their families when they might not otherwise be able to. So most might consider this to be a noble goal, but it's incredibly complicated. And one estimate was that over 200 things have to go right in the laboratory to result in a successful pregnancy. But things go wrong. Embryos may not respond well to freezing and can die. An incubator can fail. A dish with embryos can get dropped. A liquid nitrogen storage tank can leak, killing everything inside. So then what? Who's responsible? Is it murder? Is it manslaughter? And finally, many cycles result in multiple viable embryos. A couple or a person may only want 2 children, for example, but have 10 viable embryos. What do we do with the remaining embryos? So these are some of the questions that I'm going to address in the next lecture. But I hope you found this one fascinating and it's expanded your knowledge base of how IVF works.